This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 700, recorded on December 30th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here we had some pretty icky weather earlier this morning, but it's 37 now, and it's just going to turn to rain. But we were having that wintry mix for a while. So 37 Fahrenheit, which is about 5 Celsius.
It's 1 degree Celsius here. Also joining us from Austin, Texas, Rich Condit.
Hey there. 69 degrees, partly cloudy. This is 10 in the morning Austin time. And that's as warm as it's going to get today. The temperature is going to dive down to 45 overnight, 100% chance of rain this afternoon, which is fine with me.
And from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. Vincent already told you that it's 1 degree Celsius here, 34 Fahrenheit, and kind of cloudy. That's okay. Good day to be inside.
For those of you who don't know, Brianne is 13 miles away from me at the moment because I'm home. And so is she, and we're just in the same area of New Jersey, but, you know, weather could be slightly different. You never know. I think it's been snowing in Madison and not here before.
It has, yes.
The Aaron Diamond AIDS Research Center at Columbia University is looking for an assistant manager for their BSL-3 facility where you work with pathogens like SARS-CoV-2. So we'll put a link for that in the show notes. They're still looking. And you can check that out. All right, so we have a bunch of stuff for you today. This is number 700, as you heard, which is interesting because we only did 600 back in April. And usually it takes 2 years.
We used to have big celebrations when we hit these centennials, but now we're just going to move right through it.
Yeah, because I guess it's not a big deal. Go ahead.
Well, Vincent, you said we wouldn't do anything again until 1,000, but, 1,000 will maybe be in the next year, so who knows?
Well, you guys did something for 600, right? You gave me this cool virus there, which is behind me, and it was very nice. But, I don't think we need to, well, especially this is the first time we've hit the next 100 in 1 year, right? Because it usually takes 2 years to do 100 episodes. So
yes.
I suspect we might hit 800 next year. But I have to say, secretly, I've been chasing a podcast for years. It's not a science podcast. It's one of my inspirations to get me going. And, they're a weekly podcast, and they were at like 750. And years ago, I used to say, boy, I can't catch up because if we both do one a week, right? And, we're catching. I don't know why it matters, but there you go. Doesn't matter at all, actually. It's a silliness, but I have all kinds of silliness, like liking even numbers, as you may have heard yesterday on Immune. All right, so let's do a few things here. First, let's give you an update on the variants that are being discovered globally and which may be associated with increased transmission. We got another update from Public Health England, PHE, technical briefing number 2. This was sent to me yesterday by Jonathan over in the UK. And this is called Investigation of Novel SARS-CoV-2 Variant of Concern, technical briefing number 2. And this follows up on the first one, which we talked about a week ago. So that's linked in the episode from last Wednesday. And there are a couple of things here. First of all, you may remember that the variant in the UK is being detected by PCR because one of the primer sites is missing, so it's altered enough so that the band doesn't show up. So they're looking at proxy S gene target failure, right?
Yeah, I think it's that it still amplifies, but that the fluorescent probe doesn't bind. I think, but I didn't go back and double check that.
So there's basically a failure of the target gene in PCR. So they've been using that as a proxy for detecting the variant, which, isn't ideal, the absence of something, right? But they went, in this report, they sequenced 14,950 samples where they know. So now they can say, we have the sequence and what's the target failure? And they have 99.3% agreement. So in other words, the proxy S gene target failure is in fact picking up the variant because they sequenced enough to be confident of that. So if you were worried about that, that gets rid of that, I think.
Yeah, early on, there would be S gene target failures for other reasons.
Yeah.
But as this variant has taken over, virtually every time you get this particular PCR failure, it in this part of the UK, it correlates with having the sequence of the variant.
Now, they have tables showing the percent of these sequences with different weeks, beginning in October and then going through December 7th. And also they break it up, they break it up by different parts of England, like East Midlands and East of England and London, etc. Mostly, which don't make much sense to me because I'm not familiar with them. But if you're there, you know what they mean. West Midlands, Yorkshire, and the Humber. It just sounds nice. Anyway, they're very high percentages. All right, so that's one thing which I think is of interest. And then the other, which I think is important, is they did a matched cohort study to see, and they call this to inform a preliminary assessment of outcomes of hospitalization and case fatality associated with this variant. So is it associated with more serious infection or maybe less serious, right? That would show up here as well. And so the end result is they looked at 28-day case fatality, 2,700 cases. Among variant cases, 12 of 1,340, that's 0.89% cases, died within 28 days of their specimen date, compared with 10 of 1,360, 0.73% wild-type cases, the difference not significant. So it doesn't look like it's making any more serious disease, although they're going to keep looking at that, I'm sure. And it's not making less serious disease either, apparently. I guess that would have shown up in a reduction in this 28-day case fatality rate. Although maybe there are more people walking around with milder infections. I don't know. Not sure how you would pick that up. So also they looked at reinfections to see if people being infected with the variant are being reinfected at a higher frequency than if you are getting not getting the variant, and there is no difference in reinfection. So no significant difference in the likelihood of reinfection between variant cases in the comparator group. All right, so those are the important conclusions. No difference in reinfection, no difference in hospitalization, and 28-day case fatality between individuals who are infected with the VOC and wild type, well, what they call wild type. Wild type is it's a kind of a squishy thing now, right? Because they're all wild type because it's what's in the wild, but some of them are different.
I would say that wild type is anything out there that does not appear to be undergoing selection. I don't know quite. I'm not an epidemiologist, so I don't know how to describe this. But there's a background of viruses whose incidence in the population is not changing, or at least not relative to each other. These pop up as increasing in their fraction of the people, okay, relative to other viruses. Yeah, you're right. Wild type is confusing.
Because, when we work in the lab, as we all have done, we have what we call a wild type, and then we make mutations, and those are no longer wild type. And the wild type can come from anywhere. It could be a clinical isolate. You buy it from ATCC or BEI or whatever, right? So it's just a reference, I think.
Very early on, I was really clear as to, since I was going to do genetics, as to what I was going to call wild type.
Yeah.
And I spell that out in my very first paper on this. I got virus from the ATCC. I plaque purified it under non-permissive conditions about 3 different times. I grew it up, blah, 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 made a stock, and I said, okay, this is wild type.
Yeah.
Yep.
It's a designation.
If you've ever had to try to explain wild type to a non-scientist, it's really hard. In fact, I've done that in a science communication workshop exercise where different people get a word that they have to try and explain in only lay language. And one of the hardest ones is wild type.
Yeah. Now I want to see what we have in our book for wild type. Yeah, we got a definition because you got to have definitions. What is wild type? Box 3.7. Technology can be confusing.
That is
You
a true
think?
statement.
You think? Virologists often use terms such as strains, variants, and mutants to designate a virus that differs in some heritable way from a parental or wild-type virus. In conventional usage, the wild type is defined as the original, often laboratory-adapted virus from which mutants are selected and which is used as the basis for comparison. A wild-type virus may not be identical to a virus isolated from nature. In fact, the genome of a wild-type virus may include numerous mutations accumulated during propagation in the laboratory. All right, so
The wild type's the original.
It's original.
That's the lab scientist definition. The
It is.
epidemiologist definition is going to be different, and I would want to have an epidemiologist describe that to me.
Yeah.
So here, for example, the genome of the first isolated polio obtained in 1909 undoubtedly is very different from that of the virus we call wild type today. We distinguish carefully between laboratory wild types and new virus isolates from the natural host. The latter are called field isolates or clinical isolates. Yeah, so it's how you use it in the lab to distinguish things. But, you could take this variant into your lab and then make variants of it, right? And then you'd say, okay, this is the wild type and this are the variants made from it. So it's all relative. Anyway, sorry about that, folks.
The best word we came up with was reference.
Reference.
Like a reference.
Yeah, a reference. Yeah. And your reference may be different from mine.
Right.
There's a medRxiv preprint posted December 22nd.
Hang on.
Oh,
yes, sir.
Yeah.
I want to, before we move on, I want to do this now.
Yep.
You said at the top of this, you described this as a variant that may have increased transmissibility.
Right.
Okay. Are you okay with that? Because that's what NervTag concluded in their first publication, okay?
Yep.
Or their first whatever. It's not really a publication. Their first notice about which there has been significant debate, okay? They concluded, in summary, NervTag has moderate confidence that what was then the VUI demonstrates a substantial increase in transmissibility compared to other variants. Are we okay with that? I'm okay with that.
That's slightly different from saying it may. So, for example, the paper we'll look at has a way of looking at it. The CDC, if you go to their website, and WHO, they say initial studies suggest that the new variant may spread more easily from person to person. I'm okay with that, suggesting.
Okay.
I am not okay with concluding, which, as you know, the many mainstream media are concluding.
I don't care about the media. I care about NervTag. Okay? The media is a whole different question. NervTag doesn't conclude anything. They have moderate confidence. Okay?
Right.
That could be translated to suggest. They talk about transmissibility. That could be translated to spread. All they've done is track sequence through the population.
Right.
And report that sequence is increasing in the fraction of the population. They compute an R value. They compute a growth rate, okay, which is something we do all the time, okay? And they draw this conclusion. And I think it's important that this document, this group, be given the credibility that they deserve, okay? Because they've done this, as far as I can tell, correctly. We haven't seen a publication. That's not their job, okay? But I assume they've taken into account all of the, okay? But they're loaded with epidemiologists and all the rest. I assume they've taken into account all the caveats, founder effect, and a bunch of other confounding variables, and come to the conclusion that this sequence is spreading through the population more rapidly than other sequences. That's all they're saying. The rest is politics and news and the rest. And in fact, if the end result is that all the truckers get tested, that's great. Okay.
Well,
and you know.
Yes. So there are two issues. There's the science here, which is what I focus on. Then there's public health, right? So for us to get our scientific proof, maybe will take years, and we can't wait for that, right? So I agree that from a public health perspective, you say this variant may be more transmissible. So what do we have to do? Well, we have to wear masks, we have to distance, we have to reduce density, maybe close restaurants and so forth, which is great. That's fine. And eventually I'd like to know if any of these variants are more transmissible from a biological perspective. That's my position. But I'm not looking at it from a public health position. Someone told me yesterday that, we don't know if these are actually biologically more transmissible, but the epi says these things are going through the population faster, so you have to make a public health decision. And those are typically made very quickly, right? And we don't have the luxury of waiting for some experimental results. I totally get that.
The epidemiology is science.
I didn't say it wasn't science, Rich. I'm just saying it's different.
I just want to get this straight. Okay,
it's a different kind of science. It's not it's not experimentally based science. You don't do an experiment.
So to me, the kind of stuff that you're looking for, is it more stable? Do you shed more? Blah blah blah. That's mechanism. Okay, the epidemiology says it spreads faster. The next question is why? Okay, and that's where all the biological. May spread faster. That's what they say. Moderate confidence.
Yeah. And CDC and WHO are suggest that variant may spread more easily. I think I'm fine with that. I have no problem with that. What I objected to was, the press. And I know you don't care about the press, but I do.
No, I want to interject here because I think, at least for the two pieces of mainstream media that I consume, well, three if you include BBC on NPR, but NPR and New York Times, the stories that I've heard have all been balanced. They have all said there seems to be evidence that this is maybe more transmissible. Scientists want to get more data, but in the meantime, here's, what we know so far. And it's the same thing that, as Rich pointed out last week, that Tony Fauci said. Here's what we know so far about, is it more pathogenic? Do we have worries about the vaccines being able to deal with this variant? But I think that's all out there, at least in the mostly quality mainstream media. So there may be other things that, are maybe less reputable. But
the Times and NPR are not all of mainstream media, right?
True. But
very true.
And yesterday, NPR said the more transmissible variant has been identified in Colorado. That was their headline.
Yeah, so I was thinking about this the other day, last night, in the middle of the night, okay? And I was thinking that in science communication, there ought to be a word that is the equivalent of alleged, okay?
Yeah, right. That's
because when you talk about somebody who might have engaged in criminal behavior, you're legally bound, I think, journalistically, to refer to that as alleged until they're convicted. Okay? And there ought to be, maybe we could use alleged, but there ought to be an agreement. Are you listening, Ed Young? Are you listening, Carl Zimmer?
Great idea.
All you guys out there? So there needs to be a word, okay, that can be used in front of this that is the equivalent of alleged that the public understands, because they understand what alleged is, okay? And they understand immediately that means, it's not been absolutely proven, okay? We think this may be the case.
Well, I agree. I think that's a great idea, the allegedly more transmissible virus.
The problem is when you say allegedly, that has all of its own implications.
I agree.
Okay?
Yes.
And, the thing is, when you speak with journalists, they want a definitive answer, right?
Well, that's why you need a simple word, okay? I mean, and get them used to it. Say, look, some sort of scientific law. You can't do that, okay? If we tell you it's alleged, damn it. Okay, we're going to sue you if you say otherwise.
It's also like talking about a vaccine or a vaccine candidate, So
yes,
this is
absolutely.
Yes.
I agree. But so part of the issue is when, let's say a journalist speaks with someone about a criminal case, they'll talk with some lawyers who say the person's guilty and some people who say they're not guilty, right? And they provide this balanced view. And I don't know if that works with science, right? There's often no balanced view. There are people who agree with one thing and there are people who agree with another thing. And often it's not going to be resolved for many years until the experiments are done. I think it's different from law, although I agree with the alleged. There's some issues.
This is the other thing I've been thinking about, because in some of the descriptions of this, the word proof has been used. Okay, is there proof that this behaves this way? Okay, and that's a tough word. Okay,
Yes.
When is anything actually proven? Okay, I'm not sure that anthropomorphic climate change has been proven, but I'm convinced. And I can't even understand the data. I'm convinced because so many other people are convinced, scientists, okay? It's tough, but proof is a slippery one.
I mean, there's some things that are proven. For example, reverse transcriptase copies RNA into DNA, right? That's proven.
Yeah. I'll go with that.
And, before that, we knew polymerases copied either DNA to DNA or RNA to RNA. So this was, wow. And it took a while to prove it, right? But I think we're convinced. So there's some things you can prove, but this is a squishy thing because, as you say, we have epidemiology, phylogenetics, and there are other ways to explain the observations. So I agree, it's hard. But the interface with the press is very difficult in cases like this, right? And that's why some of them say it is more transmissible and others don't. The CDC and WHO try and be a middle ground and say, we're not sure yet. But in the meantime, in the end, it's all about let's vaccinate people, let's limit transmission until next year sometime when we can get everybody vaccinated. And I think that hasn't changed really, right?
Right. I think that, while I cannot parse all of the phylogenetic data, I trust in the expertise of those who have done it to tell me that there seems to be something interesting going on here. And when I've thought about some of the things that could be interesting and ways that these types of patterns could arise, there are cool ideas and room for many experiments. But I also am really glad and try to make sure that all of my friends and family know that in the end, for what, while I can spend lots of time thinking about what's happening to the RNA and are there host cell RNA editors acting on this RNA, the thing that they should concern themselves with is that the vaccine will probably still work. Their mask is still a great idea. And hopefully, they will continue with their non-pharmaceutical interventions.
I think
and
in
so
the end,
it is a difference in what we need to know.
That's the concern of people, and that is the concern of the press, is something new arising that is just going to not behave like we think it should.
Well,
right?
and I think with respect to the public health interventions, I think it is important because if this is more transmissible, then the public health interventions become even more important. And if people aren't doing them now, maybe this will scare a few more of them into doing it. But I think it could be something really serious that if it's true, and in my view of the data that I've seen, this genomic epidemiology, which I'm not all that great at evaluating either, but I trust in what some of these people are saying that this could be really serious and there could be even more deaths.
What do you mean this could be really serious? What do you mean by that?
If this is truly more transmissible, that's what I mean by that. Okay.
The original wild type is pretty transmissible already.
Sure.
And
Sure.
I think we should be masking and distancing and all that already to a great extent.
And I was just thinking today about some of those early projections that we saw last spring, and if the R naught was 3 or 4 or 5, this is a transmissible virus, but things could be even worse. So you're right; it is pretty good at what it does, but it could have some other features that could make it worse. Another thing I wanted to. Oh, go ahead.
I just in terms of transmission, all right? So this I struggle with, right? We had an amino acid change, D614G, which allegedly made the virus more transmissible, okay? And I think the jury is still out on, oh my gosh, I got alleged and jury in the same phrase. Jury is still out. Some people consider it to be, others don't. But now that virus is everywhere, so all these variants have D614G. All right, so that makes it more transmissible. Now we have more variants that have. When does this end? I don't understand how. I have never seen in any outbreak the virus becoming more and more and more transmissible. It doesn't make any sense to me. I think from backing off from whatever the data are saying, I have a big problem with that. Already we had the UK variant, and then we have one in South Africa and in Nigeria and a few other. Every country is claiming now a more transmissible variant. This simply doesn't make sense to me. That's all I want to say.
Okay.
I don't have a problem. Go ahead, Kathy.
No, you go ahead.
I don't have a problem with that. Okay, just this is what is being observed. Okay, and what happens in Africa at this point in time, at least, is independent of what's happening in the UK. They may have different R factors. Okay, it'll be interesting to see what happens when they collide. I don't know to what extent these guys might compete with each other in the environment. I don't know who's got the highest R factor. Okay, but I don't have any. Actually, I was going to say I don't have any particular prejudice. Okay, about what might or might not happen. But I have to say that we have a sort of a history, an arc on TWiV of saying with respect to outbreaks and mutants and changing in biological properties, in particular with respect to virulence, I think, saying viruses don't do that. They don't become more virulent, okay? But first of all, I'd like to revisit that, okay? I can't think of any good examples. I'd like to get a viral evolutionist on board to talk about that. And it's not obvious to me that there would be any selective advantage in becoming more virulent. We've discussed that before. But that doesn't mean that there can't be, certainly in influenza, there's selection for immune escape, okay? There are variants that show up that have different properties, and this is a new creature, okay? So I don't have a problem with this. I'm watching.
Yeah, I think that, to me, one thing that I am paying a lot of attention to in discussions like this is that it does seem that we are using the word transmission slightly differently.
Yes.
In some cases. And so with regard to sort of the phylogenetic evidence, we can see that this virus is making up a different proportion of the viruses that we find. And that is being said to be increased transmission. Is that because someone has a higher viral load, or is shedding more, or is asymptomatically transmitting for one more day? Or is that because the virus has changed in how it interacts with the immune response, so it actually can colonize easier because it doesn't have to get over some immune barrier? There are lots of things that could be the mechanism, and I can geek out in the way that I do about all of those different little mechanisms. So to me, I don't know which of those things is changing. It makes sense if a virus is adapting to a new host that some of those things could change over time. But I want to know more. But yes, there is definitely a change in frequency of this over time.
So back to your thing, Vincent, about, with this variant and that variant and so forth. I think with the D614G, at least what I've seen the genomic epidemiologists say, and some of whom are on board with it not being more transmissible, but perhaps due to a founder effect, that is chance and, being there and just going forward. But that this variant, which has the D614G in it and several others, has this constellation of mutations that seem to perhaps be giving it this ability to be more prevalent in the southeast of England and so forth. And some of the mutations, at least in the South Africa variant and in the Spain one, are in common. And so, as I said last time, where people are looking at neutralization assays for the 501Y mutation or whatever, I would like to see the data on this variant compared to the reference wild type or whatever the vaccines were directed against. So that's where I am on that. And just two other things before we move off of this PHE document 2, they do start to talk about some data with respect to secondary attack rates. So this is something where they're looking at the ability of the virus by contact tracing to then show up in the next group of infected people. And they don't do that on their matched set of individuals. So this data set is not as clean, and they're going to be getting more data on that as they can do more contact tracing and sequencing going forward. So there is that.
Got it.
And then in terms of publications, we found out last week, shortly after we finished recording, that there is a preprint from a group of people, many of whom overlap with the nerve tag group, that is posted. And I have to admit that I've only read through it once, and it's really difficult because it's a lot of this genomic epidemiology and things that I don't completely understand. And it would be really great if we could get some of those people on to talk about that. And then just one little thing, it's really kind of an aside, but it's present in this nerve tag thing. It's present in the preprint. I want to buy these people larger fonts for their figures. Oh my gosh. They have the smallest fonts, and some of them, like I said last week, it's a PDF and you blow it up and it's just all pixelated. I mean, they are like 1/10 of a millimeter high, some of them. It's ridiculous. And they could make them 5 times bigger and their figure wouldn't be any bigger. So are you listening, people? Maybe you're using R to draw your figures and you need to master the figure drawing so that your figures are more legible. Someday you will be over 45 years old and you will want bigger fonts. Okay. I'm off of my tirade there.
So we have been TWiVing for 700 episodes. Now, none of you have been here for all 700, but you've been for most of them. And, we have come to recognize each other's ways of examining issues. And as Rich said, we have developed high standards, I think, for saying a virus is more transmissible or more virulent. And, maybe those don't mesh with public health requirements. And that's fine. I understand that. However, I am not opposed to something new happening with a new virus like this. If you think I am, then you haven't been listening to me for the last 700 episodes, okay? However, I am driven by what has happened before, and you should be. That's history, and those of us who forget it are doomed to repeat it, as you know. I think science is the same. You look at what happened in the past, and you learn by it. It doesn't prevent me from seeing something new, and so that's not the case. I just have a high bar for something new happening. When I see it and the data are there, I'll agree to it. But I just want to be, what did Rich say last year? Critical thinking. That's what we need to do.
I say that every other week, I think.
I hope so. But last week it was Chicken Little, right? And so that's why
true.
I'm thinking out. That's where I'm coming from. And, I know viruses do surprising things as far as they can do things. And we're always surprised. Yeah, I get it. But your proof has to be high. And we have certain kinds of data from this, which are brand new, by the way. This whole science of genomic tracing is pretty new. And the first time it's been done in a massive outbreak like this one. And I would argue that the methods are evolving. And, for example, D614G, I had a paper in here, which I took out because I didn't want to talk too much about this. It was using computational biology approaches, and this was published before the UK variant and others have arisen, which said there's no evidence that any of these have penetrated more rapidly through the population, at least from these computational approaches. So, my view is that these approaches are in their early days and are not yet worked out, whereas, a plaque assay works, infectivity works, we know how to assay it. And so that's where I am coming from, okay?
But what about the Ebola? Wasn't some of this genomic epidemiology used there? I had this feeling that, I may be catching on to it a little bit later.
But that was a much smaller outbreak in a limited population. This is the whole world with different genetic lineages of people being infected. It's very different. I agree. Yeah, they used it to a certain extent in Ebola. And in fact, remember the glycoprotein variant arose early, and it was said to be more transmissible, and that's never been worked out. Some people find it's less pathogenic maybe in lab animals. So this is all early days. That's all I'm saying. Whereas our understanding of the biology of virus is much older and goes back farther, and and that's where my strength is. I understand infectivity and what drives it, and that's why I say I want to see that kind of information. And that's all I'm saying. But please do not ever say that I am locked into how viruses are going to, I don't want to use behave, but work, okay? I am not. I am totally flexible.
Jane Flynn on you.
I totally am flexible, and if I weren't, then this podcast should not exist, right? And
sorry.
Go ahead. It's okay. I'm done.
Just emphasize that I think this has come up several times. We need to get some of these people on the show who do this work, okay? Because we're all,
yeah, we'll get them on. Don't worry. It's just
we need to get and not
I'm
only
taking a break.
only
I'm taking a break from scheduling. All right, it's really
right. Sure, I understand. But not only these guys, but I want to talk to, kind of more like an evolutionary virologist. I think that's a slightly different breed. Somebody who can cite all of the literature about what's happened in various outbreaks and has some genomics and evolution to back that up, so that we can revisit some of these ideas about the extent to which viruses evolve and how and what the pressures are and stuff, because I don't claim to understand it.
So in terms of everything that Rich was just saying, guests with different backgrounds, to those future guests, I apologize in advance for all the questions I'm going to be asking you.
That's what we do on TWiV, Brienne. It's not something to apologize for. Come on, really. All right, one more variant-related paper, okay? And this is a medRxiv preprint posted this 22nd. Emergence and rapid spread of a new SARS-CoV-2 lineage with multiple spike amino acid changes in South Africa. I'm changing the title because they're not multiple spike mutations. For God's sakes, people, you're all scientists. And this comes from a number of labs all over the world and a lot in South Africa. And it's a different variant from the UK variant, all right? And, I erred last week in saying it was the same. So my apologies for erring. This is a different variant that emerged in South Africa under different conditions. And I don't want to go into this in too much detail because it is a medRxiv preprint, but we'll put the link so you can see it. It is quite an interesting variant, and what they conclude, while the full significance of the mutations is yet to be determined, the genomic data showing rapid displacement of other lineages suggests that this lineage may be associated with increased transmissibility. And I'm okay with suggest may be associated with, okay? That's the way to put it. Alleged. It is alleged to be associated with. The introduction has a very nice kind of summary, right? They say, we don't have a lot of, we're seeing a lot of changes in the genome as it passes through different populations. And it's infecting everyone pretty much globally, and people are all different. So you're getting different variants selected for or emerging. They say, D614G emerged, and several lines of evidence suggest that variants carrying this change have increased transmissibility. Okay. Later in the epidemic, lineages with an N439K change in the receptor binding domain of spike emerged independently in different European countries and the U.S. And they cite a preprint saying this change is associated with escape from monoclonal antibody and polyclonal serum-mediated neutralization. Now, I haven't read that paper. That's serious. Polyclonal antibody escape is potentially serious business, right? And I'm kind of surprised that a single change would do that, but I want to wait for that paper to be reviewed and published. All right. So these changes have been seen throughout this outbreak. And then they go to South Africa, most severely affected country in Africa, a new lineage, which is described in this paper, harboring multiple non-synonymous spike changes, including changes at key sites in the receptor binding domain, 3 different changes, including N501Y, which is in the UK variant, that may have functional significance. And of course, this variant also has D614G in it as well. So the paper basically goes through the analysis, the genomic analysis. They have a lovely map showing how this virus propagated through South Africa. And they don't know what these changes mean, right? We do not know what they mean. It's suggested that they may be associated with increasing transmission. But what do the data mean? They say these data highlight the need to refocus the public health response in South Africa on interrupting transmissions, not only to reduce hospitalizations and death, but to limit the spread of this lineage. And that's a proper response, I think. Now, the really interesting part, and this continues on last week's discussion, one hypothesis for the emergence is that this lineage may have arisen through intra-host evolution in one or more individuals with prolonged viral replication. Remember, that was the hypothesis for the UK variant as well. And they say this hypothesis is supported by the long branch length connecting the lineage to the remaining sequences in the phylogenetic tree. The N501 is one of several changes that emerged in an immunocompromised individual in the U.S. who had prolonged viral replication for over 20 weeks. However, they say the observed diversity within this lineage can't be explained by a single long-term infection in one person because the lineage contains circulating intermediate mutants with subsets of the main changes that characterize the lineage. Okay, so they say one would need to invoke a transmission chain that passes through multiple individuals, and so maybe multiple individuals who have prolonged replication. I think it's quite interesting, and we have a number, as we saw in the paper we did a few weeks ago. We have a number of immunocompromised individuals that are getting infected, and that could lead to the emergence. Long-term replication could lead to the emergence of these. So I find that quite interesting and needs further examination for sure. And then just to finish this out, they're doing additional work. We're performing infectivity assays, neutralization assays to see the effects of these changes on receptor binding and neutralization. We're looking at clinical data to see any changes in disease severity, and they've intensified genomic surveillance to look for reinfections. So I think those are all appropriate responses. Shutting down travel is not, because these variants are likely already in many different places. But I think surveillance, which is not equal everywhere, needs to be extremely intense. And I wouldn't be surprised if we see this kind of, as we increase surveillance in other countries, we see more examples of these variants emerging. So I think it's quite interesting. All right,
Yeah.
So finally, I noticed that if you go to look at ProMed Mail, the UK variant is now being picked up in many countries, and ProMed Mail moderator said this variant is apparently moving around the world. So I think that's interesting to see. It's in Colorado in the U.S., right, yesterday, one person. It would be interesting to see if they seed new outbreaks and take over as the dominant strain. I think that's an important observation to make.
Right. I was just going to say, and because that person in Colorado had no travel to the UK, for example, that it suggests that it's already community spread.
Yeah. And I think we said last week it's probably already in the U.S. So
Yeah. I think of it like water, if you think you could stop a flood, that's the same kind of thing with these viruses. If you think you can stop one by, not having a plane fly over from England, yeah, it's crazy.
Yeah, sometimes I wonder, we call this the UK variant because it was first described in the UK. But the UK is also doing a lot of sequencing and is very good on that front. And so we don't actually know that it started there.
Right.
Or that it hasn't gone to other places that haven't done as much sequencing.
Right.
if you look, you will find. What does it say? We look under the lamp for the lost keys, right?
Right.
Yes.
I just want to have a little sidebar here of true confession. So this use of the language talking about nucleic acids get mutated and proteins have amino acid changes. I'll confess I really only became aware of making that distinction maybe about 3 or 4 weeks ago. So it's not something that I've really paid close attention to.
No, you knew months. You knew years ago, Kathy, for sure.
I have talked about mutant proteins. I've
Really?
talked about yes.
I'm going to find an old TWiV 10 years ago where you agreed that it was the wrong terminology. I'm sure you said it.
I wasn't on 10 years ago, and I am only conscious of it for the last 4 weeks. So when you say to all these people, get it right, they may be coming along more slowly like I was. So, yeah.
Well, there are many people who don't care. And I mean, they have arguments with me that a mutation is, why can't a protein be mutated? Okay. Well, the reason is the terminology was established decades ago, and mutation is a change in nucleic acid. And if you want to change it, you have to have a reason to do so. And I think it's important because it allows you to describe things accurately. And, when you write a textbook, you have to use the same definition throughout the textbook. So.
Right. Not all of us have written textbooks, but
I will admit that before I wrote the textbook, which was 20 years ago, I used mutation and protein for sure, but the light shone and I saw the way. So,
well, I mean, and it's also, that there's a very distinct difference between susceptibility and permissivity.
Yeah.
And to people, particularly speakers where English is not their first language, those two words might not seem any different, or they might translate into their language the same. So, being aware of them and using them correctly are two different things, and you can't use it correctly if you're not aware of it. And so I think I just want to
that's fine.
cut
I
people
agree
a
with
little bit
you,
of
but
slack.
the problem is people push back and say, no, you're not right about mutation and amino acid. It's not true to say I'm not right. I mean, I agree. If you don't know about it, that's fine.
Yeah.
But then there are people who rigorously oppose it, and I don't think that's correct. But anyway, Kathy, you put a bunch of stuff in here. Why don't you tell us about it? If you want to.
No, I just had our screen of us up, so I had to find the Google Doc. Okay, so Daniel Griffin just emailed us some flu data specifically about New York State, vis-a-vis what Vincent mentioned last week about there doesn't seem to be much flu incidence this year. Kind of surprisingly, the twindemic so far isn't happening. So hopefully Daniel will keep us up to date with the New York inputs that he gets, and Vincent or we can also check CDC periodically and see how that goes. There was a story in the New York Times, the printed copy today on page A4, and I guess it came out yesterday, by Apurva Mandavili. And the first person that she quotes is Daniel Griffin. And I put the link in there, and it's easier for me to find it here. The gist of it is that they're talking about being able to use CT values to quantitate what viral loads are, particularly in patients when they first come in, because looking at that could really affect the possible outcome for that patient. If it makes sense, if there's more virus, then that patient might not be doing well. And so this article says that 2 FDA experts said on condition of anonymity, that's been the case all along, that you could use CT values. But the news quote came as a welcome surprise to some experts who have for months pushed labs to record this information. So it's also going to depend, according to email we just got from Daniel, on what kind of PCR machines you have, if they just give you a plus minus or if they give you a CT value. But
can I make a comment?
I had one more thing. It was a caveat about it, but go ahead. I'll remember my caveat.
You're going to remember it?
Yes. Yeah. The caveat is that when you're sampling, so standardizing CT values across different platforms in different laboratories, different machines, and so forth, and whereas that can be done for things like HIV where you're using blood samples, here where you're doing nasal swabs and they might be more or less, all the way back to the brain or not. There's a lot of variability there. Nonetheless, it's thought that getting the CT value information out is something that could be useful. That's all.
All right, let me show you an image of Daniel's. This is his phases of COVID, right?
Mm-hmm.
And this is kind of emphasizing what Christian Drosten told us months ago. All right, you have, in most patients, you have this huge increase in shedding from the upper tract early in infection, and it peaks, and then you either resolve or then you go on through this, the inflammatory phase. And as you can see, the viral load in the respiratory tract, this purple line, decreases rapidly after that first phase. And then when you're in your inflammatory phase, it's quite low. So I don't know how viral loads at the beginning is going to tell you the outcome, right? Because they're high. They're high, and that's how the virus is spreading so effectively, because you get high shedding from the upper tract. And I don't know how that's... I agree that CT is important for saying if you have a 35 or above, you're probably not infectious. I get that part, and for that, it's worth supplying. But I don't understand how it can be a predictor. That's all. I don't...
They're just saying... I think they're saying when you come in...
Yeah.
First off, with your symptoms. So you have bad enough symptoms that you're coming in for something about those symptoms. Mm-hmm. If they measure the virus load and give you the CT values, it seems to correlate with outcomes. And so that's what they're talking about. And you could imagine that within one hospital or, one hospital's laboratory or whatever, that over time that might start to, give you some frame of reference for that. So that's basically.
So maybe if you're in this first week, maybe there's some variability in the level that predicts going forward, right? Because that peak is not going to be a predictor, in my view. It's got to be somewhere down there.
Right. So I was just going to quote what Daniel says in this article. Tracking viral loads can actually help us stratify risk. So
I just want to see the data for that. It's an interesting idea, but I don't know what the data are.
Well, you can ask Daniel when you talk to him then.
Yeah.
So, Vincent, could you put that graph up again, please? Do you know what first of all, where's this come from? Can we have a link to this in the show notes? Or is it published?
It's not published. It's a slide Daniel has made up, and he shows, when he gives talks.
I think it might be published. I know that he has a paper.
No, it doesn't have it's not published. He doesn't put his preprints up typically, so it's not published.
Do you know what that green dotted line is? There's two green dotted lines, horizontal lines, that go across from...
I used to. I forgot now. I knew last week.
Because a comment that I want to make on all this, and this is just more a question than anything else. We've had a lot of discussion about the correlation between RNA and virus and contagion. Okay?
Yeah.
Infectiousness. And we have made statements about actually the two ends of that. Viral RNA on one end and contagion on the other end. Okay? And in order to make that connection, it seems to me that you need to, in a systematic fashion, have good numbers for how much viral RNA there is, how much virus is shed from that individual, and the degree to which those individuals transmit disease to other individuals. Okay? And, cutoffs have been drawn saying that, above a certain RNA copy number or below a certain RNA copy number, you're not contagious. All I want to say is that, like you, Vincent, I want to see the data.
Agree.
In a way, I don't doubt that that there is some minimum copy number, in particular in the tail phase, where you're no longer contagious. Okay, but I'd really like to see the data.
These might be
and know how good that number is.
Those dotted lines may relate to actual infectivity, but I will ask Daniel.
That's why I was wondering about those.
I agree, but you know what the goal would be eventually is to just use PCR as an indicator, right? If you can do the infectivity first and then say this correlates with PCR, which then correlates with outcome, that's fine. But you have to do that. I agree. I agree with
exactly. That. And in a sense, that's what people are doing, because they're saying, below a certain CT value, you're probably not contagious. Okay? And, my guess is that at least anecdotally that's true. I just don't know how good the data are, and I don't know where they come from. I know in the original Mina paper, okay, where they modeled, I want to remember that paper was really, if I remember it correctly, was just a modeling study that modeled frequent, less sensitive testing versus less frequent, very sensitive testing, and came to the conclusion that you could do less sensitive testing more frequently and manage, get as your information would be as good. It would, and in that, there in the introduction, there was a reference to 3 different papers, okay, that drew this sort of connection between RNA on the one hand and contagion on the other hand. I remember looking at those papers and thinking to myself, this isn't enough data for me. That was a long time ago. And so it would be interesting to see that reevaluated, or have somebody, write in and tell us how good are the data for that.
I mean, my concern with this article, I guarantee you within the next weeks, we're going to have people writing in and saying, is it true that CT of 20 predicts a better outcome early in infection? I guarantee that this is going to be misinterpreted, okay? And I understand. I'm not saying don't write these articles because they're interesting, but people do not know how to interpret them. And I think they are the basis, for saying that kids don't get infected, because there was a Times articles published saying kids don't get infected, right? So that's a problem. And as you say, I want to see the data, but they're not here. I don't know. I don't have a solution. Yes, Brianne.
So I have another email from Daniel that also has that same figure in it.
Yeah.
And the green line is whether or not someone is infectious, while the blue line below it is PCR detection.
The blue line, there is no blue line. There are 2 different colors of green.
Okay. Let me...
The top line is what you're called. Top line and bottom line, Brianne.
Yes, the top line is infectious. The bottom line, which is a darker green that could be blue or black, is PCR detection. Yeah.
Oh, okay. All right, good. That makes sense, I guess. But I'll ask Daniel tomorrow. Is tomorrow? Yes, tomorrow's Thursday. If it's Thursday, it must be Daniel.
Ask Daniel, show me the data.
He doesn't give me a lot of chance to, press him, because he wants to be 30 minutes because the busy clinicians,
Listen, I can forgive Daniel for lots. He's a busy guy.
When you used to come on, Rich, You didn't shy away. You just interrupted him all the time, and
yeah,
I think that's good.
Well, except that he does want to do. I actually feel a little bad about that one show in particular. I kept him rapping for 40 minutes about what his life was like. Okay, because I really wanted to know. But he's got a mission. Fine, let's let him do his mission.
Okay, sorry, Kathy. Go ahead. Continue.
Okay, yeah. Two news things that came up today on BBC. AstraZeneca vaccine has been approved for emergency use in the UK. So that's the one developed in Oxford. And so these reports said that together with Pfizer, they will have enough vaccine to cover the entire country.
By when, do you know?
No, I don't know. I don't remember. They may have said, and I didn't focus on that. It's just that they already have lined up enough, which is not something the US has done.
Does it say what dosing they're going to use?
I did not hear anything about that either. I did. The opening line in the BBC story said the UK has ordered 100 million doses, enough to vaccinate 50 million people. So that makes it sound like they're equal-sized doses, but that could be just based on old information. That remains to be seen.
So I have a question.
It's full dose. It's full dose according to this article. Yeah.
Okay.
So I have a question. This kind of gets back to our details about press coverage. People have noted that this vaccine was being approved for emergency use in the UK and have wondered, did that mean that it would be approved soon in the United States? Am I remembering correctly that there's a separate U.S. trial that is a month or so behind? So
Yes.
wouldn't anything going on with the FDA have to wait until that trial was completed and have nothing to do with these data?
Right. Something that I heard said that, yes, this doesn't impinge or make any changes in what's going to happen with the U.S. In fact, I was told that the AstraZeneca trial might still be enrolling until mid-January, but maybe that was about another one. There were two that were enrolling at U of M, and they might enroll till mid-January. So
I don't know.
Go ahead.
I don't know how these various agencies talk to each other, but I do know, at least historically, that approval in Europe does not equate to approval in the U.S. And the huge story that comes to mind is Thalidomide. Okay, if I remember that correctly, that was approved in at least the UK. I don't know how widely in Europe, and the FDA in the U.S. said, Okay,
right.
And the FDA was right.
So, if they're using full doses, which they appear, because in the article it says there weren't enough data from the half full to approve that regimen, that was only 70% efficacy, right? Which is not great.
Well, back in the day, we were going to be happy with something over 50%.
Not me.
Now we've been a little bit spoiled.
I was never happy with 51%.
I know, but that's what I think was the FDA. But,
yeah, that's right.
Now we're spoiled because we're going to see 94 or 95. I mean, what's the influenza efficacy ordinarily?
Yeah, okay. But I think it's going to be problematic with 60% efficacy.
It has the advantage that it's a lot cheaper. It doesn't have the cold chain issues.
We'll see what happens. I guess we're going to be doing 2 TWiVs a week for some time, huh?
I was going to say, if we're going to cover the world with vaccines, we're probably not going to be able to do it all with Moderna and the Pfizer vaccine.
Yeah, but I mean, I'm just questioning, right? We're learning as we go along here, right? This is all new, and I'm questioning, and I think that's what needs to be done. I mean, someone told me from Pfizer, he said, you should shut up about criticizing vaccines. It's not right. You should just let them be used. And I don't think that's right. Oh, well. Go ahead, Kathy.
So there was one more thing, and it relates to a question that we had from a listener, and I didn't zoom down to find that. But they were asking about the Sinopharm vaccine, which is one of the ones from China. And they announced today, 30th of December, that the phase 3 trials showed that it was 79% effective. And that's what I heard on the BBC. That's lower than what we were just saying about the Pfizer and Moderna ones. However, in the United Arab Emirates, which approved a Sinopharm vaccine earlier this month, they said the vaccine was 86% effective. And so that was from interim results of the phase 3 trial. So they haven't really explained that discrepancy. And
which is
so the Sinopharm is an inactivated vaccine.
Right.
Okay. And there's an article in today's New York Times about people lining up in China for vaccines for which the clinical trials are not yet finished. China has 5 vaccine candidates in phase 3 trials as of today. When, I get my New York Times that's printed, I think the deadline is 8 p.m. the night before. But this thing from BBC came out after that. So that China announced that the phase 3 trials for Sinopharm results. So
Okay.
a lot of vaccines there. That's all.
It's good. Now, the final thing is that Novavax vaccine went into phase 3 in the U.S., right?
Right.
Yes.
Just this week.
And that
And
one
so-
is protein subunit, correct?
Yeah.
Correct.
That spike with some
they-
funny aggregate, right, Rich?
Yeah, it turns out, yes. They call it a nanoparticle, but it's just because, from my understanding, it's because it associates, we'll say, I'm not going to, I think that's a better word than aggregate, by the C-terminus, okay? And my understanding is that they, when I looked into this, my recollection is, and I hope someone will correct me if I've got this wrong, is they didn't do any special modification, okay? There may be some preparation tips to get a particle size right or something like that, but this thing, when expressed in the absence of a membrane, I guess, okay, does this sort of C-terminal association that gives you this cluster with the, binding domain on the outside of the cluster. And I think that's fascinating because that's a sort of, in a way, in my thinking, sort of an ersatz virus-like particle, okay? In that you have a whole bunch of epitopes clustered together on a surface, all right? And that vaccine, being a subunit vaccine, and I have to have some long... Brianne and I need to go to an ASV meeting together, okay? and I may have to start drinking again, okay? And spend several nights in the bar with her, okay? But at any rate, that's a protein subunit vaccine. It's administered with an adjuvant, and Novavax has is, if I recall correctly, a novel adjuvant that has some... I think the word proprietary attaches to at least part of it. Okay.
We passed our paper, but let's go back to it
Yeah.
because I think it's so cool. I just think it's great. But I must say, science is another one I could not highlight in preview. I have to go to Acrobat. And all right, fine. I'm not going to beat a dead horse. Poor horse. And this is Science afucosylated IgG characterizes envelope viral responses and correlates with COVID-19 severity. And this is a large group of individuals, mainly in the Netherlands, and some collaborator here in the U.S. at Weill Medical College of Cornell. But the first 1, 2, 3 authors are first authors, Larsen, De Graaf, and Sonnenveld, and the corresponding author is Wiederson. Would it be Wiederson or Wiederson, Kathy?
I don't know Dutch.
Okay.
How's it spelled?
Vidarsson.
VID?
Yeah,
I think that's Wiederson.
Wiederson.
Yeah. When you have the two vowels I and E together, I think it's the second one that's long. So if it's EI, it's I. If it's IE, it's E. I may be remembering that backwards, but I think that's right. And if you've got just the one, I think it's a short I.
What is your
Kristen?
ancestry, Rich? Are you?
I took 4 years of German in high school and went to a German language camp in college, and so that I could pass out of the language requirements, so they wouldn't send me to Vietnam. And then came back and continued taking German, and most of it's gone except for that little bit.
I think when we were in Germany, you mentioned that.
I may have mumbled a few things.
All right, so here, this paper, let's start with a picture of an antibody molecule, a schematic. Our Y-shaped molecule that we are all familiar with, where, the top of the Y, for those of you not watching, the top of the Y is the part that binds antigen, and then the bottom part is called the FC portion that can bind receptors on cells and confer different functions. And what I really like about this picture is that it brings back our papain arc. What was that? Papaya King, the place where you can buy, juices which have papain or bromelain in them that help your digestion. But anyway, the original characterization of these two portions was made by cleaving antibody proteins with papain, long before we had any genes to look at. So the papain cleavage sites separate the FAB and the FC.
So
and
the FAB binds to the antigen, and it's sort of the arms of the Y, the top that Vincent mentioned. The FC binds to a receptor or gets in the way of stuff or does whatever function that antibody is going to have.
So today's paper is all about the FC portion and what it can do. And so that's why I show this. And again, we'll put a link to that. It's a picture from the textbook. But, they start by saying that, mostly we think about antibodies being determined by the function being determined by the top part, the antibody binding domain. But there's important stuff going on in the FC portion. In this paper, there is a sugar, a glycan attached to amino acid 297 in the FC part of IgG, and really important for effector functions. And so this is a chain of sugars. In fact, let me show another picture so you can see this in a different way. Fucosylation. No, that's the wrong one. Sorry. This is a figure from the paper where they're doing mass spectrometry to analyze the extent of glycosylation. So here's the amino acid sequence with an asparagine to which the sugars are attached. And the sugars are squares or balls. And this one right proximal to the asparagine is a fucose sugar. And that can be missing in the chain, and that's afucosylation in the title is FC without fucose, with the rest of the sugars though.
In your figure, it's the little red triangle.
Little red triangle. That's right.
So for anyone who's watching this, all of the differences we're talking about between these antibodies are basically whether or not that red triangle is present. The paper, they talk a little bit about alternative sugars that sometimes are there in the place of that red sugar, but it's really about in the place of that red triangle. Sorry, the sugar itself is not red. But it's really just about whether or not that red triangle is there. And it made me a little sad to look at this figure because I was imagining this massive change in the antibody. And then I was like, oh, I guess it's not as massive as I thought.
And remember, these sugars are added after the protein has been produced, right? These are post-translational modifications. And so whatever is regulating that's an interesting question, which we don't have an answer to. But as you'll see, there are big variations in people and viruses and how much fucose is present here. And it may make a really big difference for pathogenesis of the disease. And what's cool is that, this is part of the introduction, the discovery that IgG variants without core fucosylation, without that fucose there on the FC portion, cause elevated antibody-dependent cellular toxicity via increased interaction with FC receptors, in particular one particular receptor. But if you don't have the this is important to remember, and I kept forgetting it, and it makes you confused. So without the fucose, you bind better to FC receptors, right? No fucose,
Correct.
better binding. And so when you get better binding to FC receptors, so FC receptors are on certain immune cells, and they can make the antibodies do different things. And in particular, they call this glyco-engineered monoclonals lacking the fucose are better for killing tumors because they bind the FC receptors on some kind of immune cell that's going to then target the tumor via the antibody binding to a tumor antigen. How cool
I'm
is that?
guessing an NK cell. How about an NK cell?
Could be an
An
NK.
Is
NK
that
cell
okay?
is one of the options. Yes. So they talk
good.
about ADCC here, and ADCC is a fancy way of saying turning on NK cells.
Can be other cells like macrophages as well, right?
It can be other cells like macrophages that may change things like whether or not the pathogen is phagocytosed. That also may change the cytokine production by the macrophage.
So I think this is so cool that you can engineer without fucose a monoclonal and it'll be better at killing a tumor. This is the beauty of science, right? You figure out, oh, look, there's fucose missing on some antibodies and it has this effect, and then you use it. I just love it. So I didn't know that changes in these sugars attached to the FC of IgG differ according to your age, your sex, your disease state. For example, as you get older, a certain kind of galactosylation instead of fucosylation decreases with the advancing age. So not only are my bones getting hollow, but my IgG are lacking galactose in this position. So that's really fascinating. So people are very different in their level of fucose at that position, which is important and can control a lot of things. Okay, so that's going to be the topic of this paper, and the amount of fucose here determines a lot of disease. So pregnancy-associated alloimmunizations. When you give, there are many ways to do this, but you could give a transfusion to a pregnant woman, and then the antigens on the red blood cells can be engaged by antibodies, and you can get ADCC, which is mediated by how much fucose is present.
And this isn't usually something that happens with the transfusion in pregnant women. We're not transfusing pregnant women frequently. This is more about responses to antigens from the fetus.
From a medical article, Brianne, maternal alloimmunization, the most common routes are via blood transfusion or fetal-maternal hemorrhage. That's what it said.
Blood transfusion. Yeah, I still wouldn't think about that as happening terribly often, but I guess I could be wrong.
Well, maybe it's rare, but it may be associated with disease. But anyway, that's why I said that, because I certainly know squat about maternal allo antibodies. So this fucose is really important. And now what about viruses, right? HIV and dengue viruses those infections are influenced by fucosylation of IgG. Elite controllers, HIV or AIDS elite controllers that have very low viral loads, they are able to control the viral loads. They have no disease, basically. They have low core fucosylation of anti-HIV antibodies.
And so that implies that antibody binding to FC receptors is somewhat protective in the HIV.
One would think you could translate that into a therapy of some kind, right?
Maybe, yeah.
Although you got the problem of the latent DNA and it's always there, so you can't get rid of that. And then the other one that's fascinating is dengue, which is the opposite. So, enhanced pathology with secondary dengue infections is due to having too much. Is it too much? High fucosylation, right? And then you get, no, it's got to be low, because then you get excessive antibody-dependent cytotoxicity. So low fucose is good for HIV, but bad for dengue because you get, these are non-neutralizing antibodies which allow virus to get into other cells.
Right. So again, it sort of implies that having the antibody bind an Fc receptor is a negative in dengue, which kind of goes with what we know about ADE.
So they decided to look at IgG glycosylation in infections by enveloped viruses. All right. And they've looked at a bunch here, including, of course, SARS-CoV-2. They have HIV, cytomegalovirus, measles virus, mumps virus, hepatitis B virus, SARS-CoV-2. They also looked at a non-envelope virus, parvovirus B19. They looked at vaccination with HBV protein subunit vaccines. And they also looked at live attenuated envelope virus vaccines to see if any of those differed according to fucosylation of IgG. Really cool stuff. And they use mass spectrometry to measure the amount of fucosylation of IgG-Fc, right? They also looked at platelet antigens, because that's where fucosylation makes a difference, right? If you have, less fucose will give you better ADCC against foreign alloantigens in platelets. So fucosylation of antigen-specific antibodies against a human platelet antigen was reduced, and in a similar way, responses to envelope viruses, cytomegalovirus, HIV, showed significant afucosylation, reduced fucose of IgG. Okay, which would be good, right? It helps to clear, because you get better Fc receptor engagement and ADCC, help clear infections. And so these are virus-specific antibodies, because they say, even though the virus-specific IgG has low fucosylation, the total fucosylation in the individual is typically high. It's only specifically lower for certain situations, which is very interesting, right, to figure out how that happens. Let's see. So the extent of afucosylated IgG was variable. Against envelope viruses was variable. It varied between individuals. It varied between types of antigen. Afucosylation was very strong for CMV, less strong for HIV, so there are virus and person differences. Okay. And then they ask whether some people have a greater capacity to make an afucosylated IgG response, right? Or is some people better at it than others? Does that explain being free of disease? But so they looked at, within people, comparing the fucosylation of IgG against two different antigens within the same person, but they didn't find any people who were particularly good at it. So they say that afucosylation is not predetermined by genetics, but seems to be stochastic. It's random. We don't understand what regulates it. The next thing they did was to look at immune responses to the same antigens in different contexts, right? So they looked at hepatitis B surface antigen antibody glycosylation, fucosylation in people either infected with Hep B or vaccinated with the recombinant vaccine. It's the hepatitis B surface protein vaccine. The anti-HB surface antigen IgG fucosylation was elevated in people who had been vaccinated with the protein. And so fucosylation is elevated, which means less binding to Fc receptors, right? But in people who were cleared a natural infection, they have lower Fc fucosylation compared to subunit vaccination, right? So they say, well, this is cool because the context, either immunization or infection, is determining what's happening with the fucosylation level of IgG. And they move on to measles and mumps. Again, they can compare fucosylation with natural infection versus immunization. And here they find something different. Unlike HBV, the vaccines for measles and mumps are infectious attenuated vaccines. They have a similar fucosylation pattern in the antigen-specific IgG antibodies.
It would be so cool if one could do an experiment. One could do this experiment if you took the same antigen and delivered it with different vaccine types, say, I don't know, like one being an mRNA vaccine, to see whether different types of vaccines lead to different levels of fucosylation of antibodies, and to sort of match that to whether or not fucosylation is a good thing or a bad thing in that particular viral infection.
Well, so we're doing that experiment, right? Because people are being immunized with different kinds of SARS-CoV-2 vaccines, and then it's a matter of people just doing these experiments. All right, what about SARS-CoV-2, right? So they're looking at IgG responses against the spike protein. They found these are skewed towards low core fucosylation. However, antibodies against the nucleocapsid protein, which is not on the surface of the particle, it's not on the surface of cells, has high fucosylation. So low fucosylation, better engagement of Fc receptors, high fucosylation, lower engagement, right? So that's really interesting that the spike and the nucleocapsid antibodies have different fucosylation states. And then they look at hospitalized patients with acute respiratory distress, recently hospitalized. Their anti-S IgG were significantly less fucosylated than in convalescent plasma donors, that is, people who have recovered from SARS-CoV-2 infection. So people with severe disease, less fucosylation, which would be stronger Fc receptor engagement. Everybody okay so far? So they say the lowered fucosylation in these patients with severe COVID in the anti-S IgG suggests that this is having pathology. This is causing pathology maybe through Fc receptors, because that's the effect of lowered fucose, right, is to increase the Fc receptor engagement.
So that's a situation where, if I understand this correctly now, the increased Fc receptor engagement we're talking about, in terms of patient outcome, is not good.
That's right. And they're gonna
whereas other cases that we've talked about, it's good.
Exactly.
So
it's good.
It kind of depends, and we already said that it differs for HIV and dengue. So it depends on the circumstances.
Right. Yeah, exactly. And actually, they do an experiment to address that in a moment. So let's see, what else can I tell you here? All right, so let's do the experiment, because that's really the key observation. How these antibodies without fucose could contribute to a strong inflammatory response, because that's part of the issue with acute respiratory distress, right? And they say, so alveolar macrophages, right? These are a key cell in the lung, and they produce the Fc receptor that will bind the Fc portion of IgG. It's Fc gamma R3A is the major myeloid sensory receptor for afucosylated IgG. So they looked at the ability of these cells to produce a pro-inflammatory cytokine, IL-6. Afucosylated IgG enhanced IL-6 production from macrophages in vitro. Brianne, what did they use for these cells? Did they have a cell line that they use, or what? Do you remember? Did you look?
I don't remember. But
anyway, there's cells in culture. I'm not sure if they're actual macrophages or made from a cell line or a cell line or so forth. Anyway, if you add afucosylated IgG to these cells, they make IL-6, of course, which is a pro-inflammatory cytokine, most critical for acute phase responses in people. And they say there's a correlation between anti-S fucosylation and both plasma IL-6 and C-reactive protein in patients. So patients with high IL-6 have lower fucosylation. So that kind of plays into their idea that you have these patients with serious disease have low fucosylation, better binding to Fc receptors, and that leads to inflammatory cytokine production, exaggerated release of pro-inflammatory cytokines, and then that begins all of your problems. And I think I skipped over, or must have, but the non-envelope viruses, I guess I talked about, no, that's Hep B. They did.
Well, you mentioned them both, non-envelope viruses and subunit vaccines.
Right. So they used parvovirus B19 as their non-enveloped virus. And just to get back to your macrophage question, they actually purified monocytes from blood from donors.
Oh, good. Very good. That's good. So the thing is, these effects are seen with membrane antigens, not soluble proteins like from parvovirus B19. And so the question then is, how does this work? And they have a model, which is a way of let me see if I can show this to you here. I think I brought it up. Fucosylation. No, that's the wrong one. Here we go. Brianne, maybe you would do a better job at explaining this.
Okay, so in the first part of this figure, in part A, they are showing just the antigen binding to the B cell receptor.
That's this little purple thing here, right?
Yes, that's the little purple thing.
Purple lollipop.
Purple lollipop. And signaling to that B cell. We can see that with the case of a red blood cell, we have this membrane-bound antigen. And we also have the ability of some other sort of additional receptor, this self-cosignal receptor, that is binding to help give the B cell a different signal.
Yeah.
That's potentially altering what's going on with the fucosylation, and you might see a similar interaction happening with an infected cell. You could also imagine, it's not really shown here, the way that the antibody molecules could bind to FC receptors and that influencing what the macrophage or NK cell does.
Yeah, so the cool thing about this is that the data with red blood cells where, it's a membrane protein, the alloantigen is a membrane protein, led them to think, well, infected cells have viral membrane proteins in the plasma membrane, so there must be some commonality. So that's why they're postulating this second interaction with the B cell. And how that affects glycosylation, of course, we have no idea the mechanism there, but...
Right,
it's a lightning bolt.
A lightning bolt, yeah.
Exactly.
Yeah, see it?
Yeah.
It's a green lightning bolt. So importantly, the self-co-signal and the self-co-signal receptor, that's all hypothetical.
Yeah.
It is.
Right?
They sort of postulate such a receptor existing, but they haven't come up with it. They haven't identified any particular receptor or anything like that. So, they say at the end that they're hypothesizing this receptor. Yeah. It's certainly one part that, gets me as an immunologist, sort of like, oh, I really want to know more. And there's another part in the intro, the sentence that frustrated me the most about the mechanisms controlling IgG core fucosylation remain unclear. And then they just kind of moved on. And I was like, no, I want to know how it happens. Mm-hmm. That's the part I want.
So what I really like about this is that the model explains why a membrane-bound antigen behaves differently in this than a soluble antigen, because it's only with a membrane-bound antigen that it can be in conjunction or proximity to this hypothetical co-receptor, whereas in a soluble antigen, that's not going to happen. And I think the beautiful internal sort of control for this is the difference between the spike protein and the N protein. The N protein would be a soluble antigen, and you get fucosylation, and the spike protein would be a membrane-bound antigen, and you get afucosylation. It's brilliant.
Right.
It's nice.
And I like the same experiment with the hepatitis B as a protein subunit, as opposed to being delivered by natural infection, also giving that nice control that it's about the context of the antigen and not the antigen itself.
Yep.
So what I found really interesting, the last part of the discussion, they talk about using convalescent plasma to treat people, right? Because depending on how much fucosylation there is in that particular person that you get the convalescent plasma from might make a difference, right, in the outcome in the recipient, right? So they say our observations have direct consequences for convalescent plasma therapy. We should use plasma enriched in fucosylated anti-SARS-CoV-2 antibodies to avoid escalation of symptoms and promote virus neutralization in patients. And that seems so clear. I mean, some of these convalescent plasma trials have been associated with issues in recipients, and maybe fucosylation was part of it. Maybe they need to look at that. Or maybe you just need, and I thought even beyond that, the monoclonal antibodies that are being used, I don't know what the fucosylation state, maybe they should make sure that they're fucosylated, right? Or, to promote neutralization and avoid the ADCC, right?
Right, exactly.
And the other thing that occurred to me is that we know that people mount an immune response to a protein subunit vaccine that's different than an immune response to a live vaccine. And I wonder if this isn't part of it, though. Now, was any of their controls? No, we don't have an inactivated membrane-bound vaccine in this because.
No, they don't.
Okay, so there's more to know about here.
That's why I would love an experiment where they took the same antigen and delivered it as a live attenuated and as an inactivated and as mRNA and as protein subunit and did the sort of comparison to see which ones gave you fucosylated antibodies and which ones did not.
Now they go back. One final point I want to bring up. They say in their discussion, they go back to Stanley Perlman's paper on SARS-1. They say attenuated vaccines varying spike proteins of SARS-CoV are known to produce strong antibody-dependent enhanced responses mimicking pathologies in critically ill SARS-CoV-2 patients. This suggests subunit protein vaccines may be a safer option as seen in rat models for SARS-CoV-2, unless the vaccine also induces a strong neutralizing effect. So I think this is fascinating because the mRNA vaccines are going to make a membrane-bound spike, right? But that may be okay because they're so protective, right? They have 95% protection. So maybe, as they say here, they're strongly neutralizing, so you never get a chance to have ADCC, even despite the fucosylation state. Because if it's membrane-bound, the idea is going to be you're going to have reduced fucosylation, and that could be bad, right? But if you block reproduction early on, it's not an issue. So they're saying that protein subunits may be a safer option, but because they're not going to be membrane bound, right? But the mRNA,
but
right?
It's not all just afucosylation.
Of course. No, of course.
It's
other things as well. But I think it's a really interesting part of it.
Oh,
right?
Yeah. No, it really is. I still want to know where the fucose comes from.
Yeah, it's another piece of the puzzle that we have to throw into the mix.
Fucose. So this is how we learn fundamental stuff from a pandemic, and it can be applied. And I think this idea, I don't know if people are still using convalescent sera, you got to check because it's pooled, right, from different donors. And so, I don't know, do you check each donor for fucosylation? I don't know. I think you just throw it out and you make monoclonals that are fucosylated, right? You want fucosylated so they don't make ADCC. Yeah. Okay. So that's enough of hardcore science for today. Let's read some poems.
I'm exhausted.
But it's cool, isn't it? Isn't it fascinating?
It's still exhausting.
We had poetry submissions. We're going to give away copies of Principles of Virology, the 5th edition, where you could read about wild type and mutants and so forth for yourself. So we had many poems submitted. Gosh, I didn't even count them.
Oh, I was hoping you were going to.
I think it was over 50.
There were a lot.
Well, we're going to
6 pounds.
Did you do the calculation all within that spreadsheet, or because?
Yeah.
Okay.
Yeah, I put them all in the spreadsheet. I will actually publish every submission in the show notes. But we're going to just read like 5 of the top. We all ranked them by numerical scores. All the members of TWiV, and we had one clear winner with the best score that was statistically well separated from the others.
I was going to say, did you throw out the high and low score and do a standard deviation and test for statistical significance?
No, you want to do that? You want me to do that?
No. Being facetious.
Well, and based on the fact that I noticed that some of my scores were not in the right line, and I had to go back and put them back in. I wondered whether you
I
tested
checked to
for
make
whether
sure.
you got everybody's score, and everybody scored
that
everything.
was one thing I looked at, because I have a spreadsheet, and I'm just doing a sum, which wouldn't work, of course, if some were not scored. And so I
Right.
made sure they were all scored. And Dixon had put his scores in some wrong places, so I fixed those and so forth. So I think I got it right.
Well,
Okay.
based on the point spread that you're reporting here, the number one is a clear winner.
Number one is
Yeah.
a clear winner, and I think we'll give
I
away
think the
some
runner-up is pretty much, well, yeah, it's hard. They start to cluster after the first
Yeah.
one.
I mean, I don't have an infinite number of books to give away, but let's read the first one. I mean, does anyone have a particular desire to read the winner? I mean, I'm happy to let someone else read it. Otherwise, I can read it. Okay. So the first one is written by Lawrence, and it's called SARS-CoV-2E, with apologies to Lewis Carroll. 'Twas COVID, and the aerosols did spread the virus far and wide. Pandemic yielded high death tolls as obese old folks died. Beware the SARS-CoV-2, my son, the spike proteins, the RBDs. Beware Great Barrington, and shun 5G conspiracies. He took his mobile phone in hand, downloaded contact tracing app, and what is more, his mask he wore, held on by rubber strap. The R value is 2.5. We need to get it less than 1. What we must all do to survive is emulate Taiwan. Attenuated subunit, vector or RNA vaccine? What drug to use? The wise don't choose hydroxychloroquine. And hast thou slain the SARS-CoV-2? Not yet, but we can do this task. Test every day, lateral flow assay, he answered through his mask. 'Twas COVID, and the aerosols did spread the virus far and wide. Pandemic yielded high death tolls as obese old folks died. And then, so that's the main poem, which I think is fabulous. Then he has an alternative verse 2, which he writes is less political, more virological. Beware the SARS-CoV-2, my son, the spike protein, the RBD. Beware Ebola too, and shun the influenza B.
What I especially
I
about
like his
this,
original. Sorry, go ahead,
yeah,
Kathy.
no, that's fine. Yeah, I think I like the original too. It's not too political. What I especially like about this one is that it scans, and he clearly worked to make it scan. So, for instance, the test every day lateral flow assay. He and I'll just tell the story. There was a faculty member in my department at University of Georgia who liked to write limericks, and he had an exhibition of them up in the ecology building once. And a friend of mine, not a scientist, and I decided we would go read them all. And it drove us crazy because there was not one limerick that scanned correctly. And we would both stand there, and within a minute, we could fix each limerick by changing a word or a syllable or something. And so I rated these poems a lot on how well I thought they scanned, and this one was, and the content is really good, but it also scanned well.
I agree, Kathy. I didn't know the word scanned, but now I do. And it's almost like some people got a better sense of rhythm than others, or something like
Yeah.
that. I don't know, but I have to say that this is. I've recently, actually, some time ago, I never followed through with this. Tried to memorize the Jabberwocky because it's brilliant. Okay, and this is just absolutely brilliant. I have to say, Vincent, you're reading it. This is corny, but it literally brings tears to my eyes.
Yeah, no, it's fair. It was
It's
very
brilliant.
It's really good.
Absolutely brilliant.
So, Lauren, send your address so we can mail you a book, TWiV@microbe.tv. And we have some others who may or may not get a book. We'll see. Kathy, can you read the next one?
The next one, The COVIDiot, a parody of Edgar Allan Poe's The Raven. So you're going to see a theme here that there are a lot of parodies or things written in particular poetic styles. So I'm not going to do as good a justice to it as Vincent did, but linking Google Scholar queries, I grow frustrated and weary, debunking myths that I have gone over 1,000,000 times before. Still I try to make it through, but I have better things to do than argue and debate with you who has never listened to me before. They'll listen this time, I hope, posting articles which they can't ignore. The data wasn't this good before. Quoth the COVIDiot, I need more. Then you ask me to explore an article over which I pore. This is proof of a grand scheme. Politicians are ruining everything. Not so fast, I do exclaim. This article is really lame. It has confounders galore. With just Plaquenil, the outcome's poor. Quoth the COVIDiot, I need more. I know you think you are immune, but it was only back in June that so-called herd immunity was impugned. Talking to you is a chore. I thought you might have got a clue, but your new proof has an N of 2. Masks are not an evil plot. Interpreting science can be taught. Quoth the COVIDiot, I need more. Then the denier does present a letter from the president. The pandemic's over, he does proclaim. Everything is normal again. But cases and hospitalizations rise. Can't you believe your own eyes? We've gone over this many, many times before. Quoth the COVIDiot, I need more. That was by Amanda.
It's also
Brilliant.
awesome. It's really good. As Dixon said, we have quite literary listeners here on TWiV because they were all really good.
Mm-hmm.
Rich, can you take the next one?
Pandemic prophecies, pandemic snafus, pandemic prognostications, pandemic blues, pandemic rags on our faces, pandemic 6-foot distance spaces, pandemic truths, pandemic lies, pandemic politics, pandemic flies, pandemic loneliness, pandemic alliance, pandemic expertise, pandemic science. Pandemic vaccines will come at last, and this pandemic will be in the past. That's by Eugenia.
Nice.
Beautiful.
See, it doesn't have to be long. Brianne?
Yes, I'll read the next one. And it is another sort of take on a famous poem. And this one only uses 18th century vocabulary as well. It's called The Virus. Virus, virus, binding tight to our tissue day and night. What immortal hand or eye could frame thy fearful symmetry? And what bellows and what wants could twist thine host's primed response? A gentle furnace, why require such sight unkind, a storm of fire? Whence thy bloom, pressed to select a deathless force to infect? What dread hand, and who to blame, as thou on parting spark host to flame? What the template, what dread grasp, as breath gives way to final gasp? Did she smile her work to see? Did she who blew spirit make thee? Virus, virus, spiking tight to our tissue day and night. What immortal hand or eye dare frame thy fearful symmetry? Yeah, that's really symmetry, but
And virus is spelled VYRUS.
yes.
Very cool.
And so is the original poem's title spelled in an odd way.
Tiger.
The Tiger is Tyger by William Blake. So,
All right.
And
then we
I-
have,
They
go ahead.
also say the sight unkind, a storm of fire line in there is supposed to be a reference to a cytokine storm.
Ah, cool.
yeah, and as I started reading this, it was like, I know this. This is ringing a bell with me, and it was it was because it's part of the one of the poems that William Bolcom put to music in the Songs of Innocence and Experience 3 CD musical work that I was a part of, and so it's very cool. Well done. And who? This one doesn't.
There-
have the
there was no
associated
name.
with it.
Oh.
Yeah.
A virus. I'll have to. VYRUS. All right. When someone's reading, I'll look for it. Next one is Sonnet 19. Shall I compare thee to a new vaccine? Thou art more timely and more desired. Rough winds do shake this gloomy autumn scene, and summer's hope hath not yet transpired. Sometime too sore the injection sites burn, and often with some redness and swelling. Headaches and joint pains will sometime return with fever, fatigue, and Racanielling. But thy immune system works hard for thee. Every B cell and T cell now is made to save your life, so very dear to me, that death not brag thou wanderest in his shade. Embracing science and technology, will have vaccines, and these give life to thee. And it brings back my Shakespeare days, my gosh. It's by Mike. Kathy.
I was just looking up. Looks like the virus was by Peter.
Peter. Yes. Okay. Thank you, Peter.
So now I have to go back to this other document. And this is You Can't Be My Neighbor
Yep.
by
Yes.
by Nancy. With apologies to Fred Rogers, Won't You Be My Neighbor? It's a virus-filled day in the neighborhood. Another lousy day in the neighborhood. Just stay inside. Don't go outside. It's a pandemic across the USA that makes it impossible to play. Just stay inside. Don't go outside. I've always wanted to live in a neighborhood like this, one where you can't shake hands and never kiss. So let's wash our hands every day and wear a mask when we are away. Just stay inside. Don't go inside. Don't go outside. You can't be my neighbor. Never outside. Stay inside. You can't be my neighbor.
Nice. I wished you would have. I was hoping you were going to sing.
Yeah.
But maybe that's because I grew up too much on Mr. Rogers.
Right. And I wasn't sure I could put all the syllables
Is
to the right.
the rhyme like it's a virus-filled day in the neighborhood? Is that something like
Another
that?
lousy day in the
It-
neighborhood. Just stay
Yeah.
inside.
Okay,
Don't go
got it.
outside. Yeah.
Rich, could you take the next one?
The year of 2020 has been an odd one, if truth be told. It has made us think, it has made us fear, and for some, it has made us old. We try to play nice, we try to play fair, and heed the advice with an open heart, but misinformation, for many it seems, has become something of a special art. Let's Twitter this and Twitter that, propagate memes from me to you. I liked and shared it, didn't you see? Therefore, it must be true. The words that we dislike the most are the ones that end in emic. Epi soon turned into pan, and then, oh help me, came polemic. Words are flying and tempers are roaring, all while the case rates soar. Politics is taking over, and really, it's become something of a bore. It's November now, and look around. Things still really aren't that fun. We haven't conquered, we haven't won. In fact, we've only just begun. So as the season closes in, let's try to do our best. Let's be friendly, let's be thankful, and considerate of the rest. Our planet with its communities is a truly beautiful place, and I know I can protect it because up my sleeve I hold an ace. I'll wear my mask, I'll close my door, and keep myself locked behind. It's for your health, not just for mine, that this is the truth I think you'll find. Very nice. Katie
Cool.
Jane Shank.
Katie also sent in a recording
Shannock. Yes, she sent a
of her
recording.
reading.
Yeah.
So she
Her
was very
reading
cool.
is very nice. Yes.
Who's next? Brienne? Yes.
Yes. So this is from Andres. Preaching scientific fact, endless banter, but does not detract. Do not allow the paranoia to fester. All mutations are near negligible. Ask your professor. Now listen up. This is for your protection. Try not to confuse the virus name with the infection. It needs to be said, I don't want to be mean. Call it SARS-CoV-2, not COVID-19. And if you look at the first letter of all the lines going down, this one spells pedantic.
I forgot to bold them. Yeah. Andres had bolded them in his original, etc. Pedantic. Oh, this is great. This is just great. I only have 5 books to give away. I have to see how far down we got. but this is good. All right, next one is by Jen. Some viruses can make us sick and some can make us strong. We never seemed to mind too much till COVID came along. Into our homes, behind our masks, our future is quite vague. Let science be our beacon now to guide us through this plague. Let science be our beacon now to guide us through this plague. Kathy.
SARS-CoV-2 Sestina. Dear coronavirus, welcome to the human realm. I hear you're new to these parts, that you've traveled some distance to be with us today. Do you need a nap, a map, a live host to settle into? You've leapt from world to world. I bet you'd like to catch your breath. Years ago, we met a pathogen who really took our breath away. Influenza type A, quite a virus, I recall. Maybe you know her? She toured the world on her first trip. You plan to too? We knew you might. We hoped you'd not. Like you, we want to live long, happy lives. We want to go the distance. So please forgive us if we choose to keep our distance from your cruel spikes, your stunning crown, I mean. Breathe in, breathe out. That's our fuel. That's our life force. It must be different for a virus, because you're not alive, you say. Of course. I knew that. I forgot. Still, you're a wonder of the living world, an animate inanimate. Right now, the world's most ruthless replicant, too small to see. Like your distant cousins, dengue, hanta, variola, no longer new to us, but no less clever, shared silently by breath or blood or bite. Still, stealth you have, dear virus, such superb contagious skill. But since you have no life to live, I have to ask, why are you here? To change the lives of living things? To teach us? To undo us? We have a world of questions. Will you answer? A novel virus doesn't visit every day, and most are so aloof and distant. It's true. Well, I won't waste my breath on them. I'll just say that very few can spark a new pandemic, steal every headline in the news, halt our hearts, and turn our lungs to glass. Our lives depend upon your arrest, I'm sad to say. We'll all breathe better once you've passed. Is there no mercy in your world? How could there be? You have no distant friends you miss, no fear, no death? You're a virus that's gone viral, a wily rival not alive. A piece of bad news. Keep your distance. A world enveloped, born on breath. And that's by Allison.
Anyway, there are many more too.
I
awesome.
didn't notice the first time through the anagram of viral and rival. So that's nice.
Okay.
Yeah, that's gone viral, a wily rival, not alive.
All right, so I have 5 copies only. So, of course, the winner was, so that's Lawrence, TWiV at microbe.tv. And also, please, Amanda, send your address. And Eugenia and Peter. Mike. Send your address to TWiV at microbe.tv, and 5 copies shall be in the mail. And we'll send you the books, and all of your entries will be on the show notes, microbe.tv/TWiV. But they're all great, wonderful, and we've done poetry contests before, but we got so many more submissions. It's great because we have more listeners.
So
I was very impressed. I don't know how you all were able to do it.
Yeah. Vincent, you have a specific site for TWiV poetry, so these will go there as well as in today's show notes.
Oh, yes. So they will. So what happens is I put them in the show notes, and then Jolene will copy them from the show notes into the poetry page, which is microbe.tv/poem.
I think it's TWiVerse.
TWiVerse, yeah. That's right. I have forgotten. TWiVerse. Yes. And she adds them, she scans through the show notes and adds them to that. So the last one was Mark's from last week. She's got it. So
Excellent.
They'll all
Thanks,
be in
Jolene.
the TWiVerse, yeah. But I must say they're all fabulous. So thank you all so much. Okay, let's wrap it up with some picks. Brianne, what do you have for us?
So I have the book that I just finished reading. It is called Ready Player 2. It is a sequel to a book that people may have heard of called Ready Player One, which was also made into a movie. They're sort of futuristic. It's a sort of future world where everyone lives in a video game simulation. And I will admit that I reread Ready Player One in preparation for Ready Player Two coming out, and it read somewhat differently this year after the amount of time I spent on Zoom than it had the first time through. But Ready Player Two is really interesting because the characters are able to experience one another's lives through the video game, and so it really makes you think a lot about empathy. And it was just a fascinating read. So if anyone is looking for a somewhat sciencey read that doesn't focus on coronaviruses and lets them take a slight break from all of the other things we've been doing, this was just a fascinating book.
Wow.
Cool.
That's cool. Rich, what do you have for us?
Just a quickie here, something I found on Facebook from it comes from a website called Phosbites, which is a tech website that has a Facebook page as well. And this is just a sort of a montage, if you like, of robots from a company called Boston Dynamics, showing what they can do. They're running, opening doors, etc. They're robots that look like people, robots that or behave like people, have arms and legs, robots that behave more like four-legged creatures, and etc. And it's just amazing. Okay,
It's really hilarious too,
with really cool music that goes with it.
Yeah,
Very fun to watch.
They even have one that can load the dishwasher.
Yeah.
I loved it. I laughed out loud when I saw that.
Actually, the one I think I liked best that was sort of the creepiest was the doggy thing that opened the door.
Oh,
okay,
that
because
was
it pulled down the
amazing.
handle and pulled the
Yeah.
door back, and then even stuck its leg behind the door to keep it from closing.
Yep.
While his buddy robot dog went through.
Yep.
Unbelievable.
Yeah, it was really impressive.
Yeah. Someday they're going to do everything, right?
Yep.
Yeah.
They'll replace us and they won't get viruses, at least not biological ones.
Yeah.
Kathy, what do you have?
I picked something that was published on December 25th. It's a blog post, Bert Hubert's Reverse Engineering of the Pfizer-BioNTech Vaccine. And he has written a number of things, evidently, aimed at computer people. But he does the best job of explaining all of the RNA sequence that's in the vaccine. So he puts the sequence out, but he does it, and he describes it in terms of coding types of things. For example, he starts at the beginning. And he also gives like drug names for this thing. So it's tocilizumab is this, or Comirnaty are two of the like druggy names for the vaccine. But so he gives the link to the digital code, and now I'm trying to find the part that I did that I like the most. Oh yeah, so when he's talking about stuff at the 5' end, he says it starts with two nucleotides, GA. This can be compared to every DOS and Windows executable starting with MZ, or to Unix scripts that start with #bang. And so he does that all the way throughout in terms of describing the various aspects of the sequence. But, he shows how there's not a single U in the sequence because they're all this modified U, and he explains why. And then he goes through and he explains the modifications to make it more translatable, putting more G's and U's in. And then he describes and part of the signal peptide and the modification to put in the two prolines that stabilize the spike. And it's just brilliant. Like I learned things from it, and I think the computer people will learn things from it, and I think the biologists will learn things from it. So I highly recommend this. There's a part too where he talks about this modification. He said that to make it the pre-fusion form, the people that discovered this should be walking around high-fiving themselves incessantly. Unbearable amounts of smugness should be emanating from them, and it would all be well-deserved. And then he says, update, I've just been contacted by the McClellan Lab, one of the groups behind the proline discovery, the one that led to this stabilization. They tell me that high-fiving is subdued because of the ongoing pandemic, but they are pleased to have contributed to the vaccines. So it's just really cool. Take a look at it.
It's very cool.
Beautiful. I need to read this.
Yeah.
So I want you guys to tell me what the 10 nucleotide insertion is in the poly-A sequence. If you haven't thought about it,
yeah, so,
that's fine.
Well, yeah, so he says he couldn't figure out what it is and surmises that it has something to do with proprietary optimization. And I didn't even look at it in detail.
No, I think there's a specific function of it. Do you know, Rich? These are made by T7 transcription, right?
No, I'd have to look at it exactly. But without even looking at it or anything else, my knee-jerk reaction is that it might have to do with stabilization. But I just don't know.
All right, take a look at it. We'll discuss it on Friday, okay? I think it's very cool, and you'll be. You can figure it out. It's T7 transcription, and remember, they have to have a poly-A signal, right? Because this is going to be polyadenylated in a different way that the spike mRNA usually is in a virus-infected cell. So it's a DNA-based polyadenylation. But it's cool. Think about it.
I wonder what happens if you just Google the inserted sequence.
You get some references, but people are trying to figure out what it does. They don't actually know.
Okay.
Check it out. It's fun. My pick is Fever by John Fuller. Here it is. It's my original copy. It's a paperback from 19 what? Let's see. It was published in 1974. And I've had this, since I graduated college. And as I said, this was my inspiration to do virology. I was just looking at it. It's by John Fuller, The Hunt for a New Killer Virus. It's about the emergence of Lassa virus in Nigeria in the 60s. And they have a quote here on the front page. It's by Michael Crichton from the Andromeda strain. But things are now under control, Stone said. We have the organism and can continue to study it. We understand what's happening now in terms of mutations. That's the important thing, that we understand. I love it. It's just great. Anyway, this was the topic of TWiV number 9, which was recorded on November 22nd, 2008, Dixon and myself. It was called Fever, the Discovery of Lassa virus. And here's the description. Vincent and Dick recall the discovery of Lassa virus in Africa in 1969. A non-fictional account of the story Fever, written by John Fuller and published in 1974, inspired Vincent to become a virologist. Part of the story took place at Columbia Presbyterian Hospital, now Columbia University Medical Center, and now Columbia University Irving Medical Center, where both Vincent and Dick are employed. Dick remembers many of the key players in this medical drama. Now, you may be wondering why it's Dick. I called him Dick for many years because he used to say, "Hi, I'm Dick Despommier." And then all of a sudden he said, "I don't like you calling me Dick anymore." Oh, okay. Well, what do you, so Dixon. So to this day we call him Dixon, but he in fact used to say, "Hi, I'm Dick Despommier." So I never got that. But anyway, the book is the key. It's really cool. And it's a great story because it's not just about the outbreak, but it's about Rosie Casals. No, not Casals. She's a tennis player. The nurse, Penny Pinio. Nurse who got sick in Nigeria. She was airlifted back to Columbia. They took care of her. She recovered. They saved her serum. And then her serum saved Jordi Casal's life, a virologist at Yale who was working on the virus. He contaminated himself. He happened to live in Washington Heights. He commuted to Yale. So they put him into the hospital. They gave him her serum. It's so cool. There you go. Great story. Yes, I read it and I said I have to be a virologist, but here we go.
Well, I subsequently read it on that recommendation. I don't know if it was right away, but ultimately it's a great book.
Did you like it? Yeah.
Oh, yeah. The one bit I really remember, just because it's a closely familiar name to me, was there was a little vignette on Tom Monath, who,
That's right.
if I remember correctly, he was responsible for identifying the animal host.
I also should say the reason I'm picking it is because you can get it as a Kindle now, and that TWiV, there was only it was out of print, and you could only get old editions, which are very expensive, but you can get it on a Kindle now. And I remember years ago, I did some event at the Strand bookstore in New York. I think Stuart Firestein and I did something in front of an audience where we talked about viruses. I think it may have been during Zika. I don't remember. But afterwards, one of the members of the audience, she came up and she said, Professor, that book you like is over there on the shelf. So the event was on the rare book collection floor of the Strand bookstore. And she said, it's over there. And I went and looked, it was $150. But she remembered from TWiV 9 that I liked that book. And she said, go over there. And it was on the same floor that we did this event. So cool. All right, show notes, microbe.tv/TWiV. Questions and comments, TWiV at microbe.tv. If you like what we do, microbe.tv/contribute. Rich Condit is an emeritus professor, University of Florida, Gainesville. He is currently at Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.